We're going to turn now to looking at some animal models of carcinoid nets. And for this, I have the pleasure of introducing my co-chair, who's already spoken to you. You all know Girish Shah, who wears so many hats, both being on the board, being a chair, being a speaker here today. I wasn't aware of the fact that he actually wears two hats at work as well. Girish does research not only on neuroendocrine tumors, which is how I know him, but also on skin cancer. And so he comes to us from the Laboratory for Skin Cancer Research, but I don't think you're going to talk about skin cancer today, are you? So let's hear about opportunities to examine therapeutic options in these animal models, Girish.
Thank you very much for that kind introduction. No, I will not speak about skin cancer. So what I'm going to talk about is animal models of carcinoid NETs. I have worked for last 4 years or so, since 2001 actually, more than 4 years now, on carcinoid patients coming from Calgary and from New York, and we landed up making certain discoveries, such as those who are suffering from carcinoid syndrome also have a tendency to develop niacin deficiency. So this turned out to be a switch, as if you would like to say. I was working with niacin deficient mice and skin cancer, and I landed up meeting Dr. Warner's team in Washington, and they led me to this completely fascinating field of NET research. So we eventually published that paper, and since then I have been constantly thinking, how can I apply this discovery that some of the NET patients would be suffering from subclinical niacin deficiency because of serotonin overproduction. I won't go into the detail because I had spoken about it at earlier time, and we have published this paper. So if you want, I can always give you more detail. For me, I am an experimental cancer specialist. So really for me, I cannot work with cells alone. That will not give me the full picture. We try to reproduce everything in an animal model because that is the closest we can go. We can do a lot of analysis, we can play with the genes, we can do chemotherapy, and of course it's easier to get approval to do animal study than to go to the ethics board in my hospital and get any approval for the human studies. So I have done both aspects of it, and I prefer the animal model. They eat what we tell them to eat. They are not going to take vitamins or whatever just the day before they visit the clinic. And therefore, I think it's as you can understand, there are advantages of animal model. But it all begins actually, all the animal models, and I'm sorry if the colors don't come out in this lighting condition. I thought I was being innovative by putting different colors, but let me walk you through that. Essentially, for last 35 years or 40 years, people have been trying to pick these tumors from patients, suspend them in some special media, and try to isolate a cell. The general idea is simple, that if you get a cell that is isolated from a tumor, and then we can work with these cells and tell what can work or not work with these cells, what treatment will work, what is wrong with this cell. So yes, it is different, it is removed from the patient, and therefore there are bound to be changes. Cells are not original tumor that was isolated from the patient. But nonetheless, that's the closest that we can go. Advantages are there. You can use these cells and make similar looking tumors in animals. So what are the cells that have been isolated? And this is just a selection that I'm giving you. If you would look at the foregut carcinoid, midgut, or the hindgut carcinoid, foregut has, these are the number of cells that have been isolated since 1984. I have marked some of them quite clearly over here in red because I think that a lot more work has been done with them. And one particular cell line that I will refer to you over here is BON. It's a pancreatic, well, not really. For that also, I think we can have a debate on this issue. But it's the group of Mark Evers and Courtney Townsend who actually published with this. So this was an original. The primary is not known, at least as far as I know. The secondary also, I don't think it is the metastatic, but it is the lymph node near the pancreas where they recovered these cells originally from the patients. It does have a lot of characteristics typical of NET cells. Lots of groups have used it. It has been passed on from labs to labs. They have been modified, but to a large extent, a lot of work being done with bone cells is a good model. There is quite a few in some of the data with bone cells in my lab. I will show you too. Midgut carcinoids, if you look at this, there are also, I'm not going to read through all the series, but some of the important ones that I'll highlight to you or I'll point out to you is KRJ series, GOT1 and the CNDT2. So this is the Lee Ellis group, which recently published in 2007. In fact, midgut carcinoid being the majority of the tumors in carcinoid patients, there is a lot of work that needs to be done, but there is dearth of cell line. So this is the group that published in 2007 that we got the the midgut carcinoid called CNDT2. I will talk a little bit more about all these cell lines. There are advantages and disadvantages of each one of them in general sense. And hindgut carcinoid, we have just this particular one. Some studies have been done. So if you ever wonder what is the water cooler discussion we do, this is the kind of discussion we do. I'll give you, you are a privy to this discussion, the way we do it. So we have been working with CNDT. I've been working with bone for about 4 years and CNDT for about last 2 years since they were discovered. And then suddenly we find results which are not right, and I'm not the only one. Some of my colleagues have also told me that they found results which were different. And eventually now we know that CNDT is not really something that actually came from the patient. So when they tried to go back and say, okay, this cells I got from this tumor of the patient, is it same or not? And they didn't find it. And now everybody is trying to rush around and identify, is this a good cell line? The developer of the cell line, of course, stands by it and says it's a good model. The rest of them say it's not a good model. But this is just one example of water cooler discussion. But more than that, the fight also spills out in literature. Sometimes it's very overtly, sometimes in a slightly concealed manner. But for example, the bone cell line, the authors have recently published in 2008 a very nice article saying that our model is very good. It also provides the symptoms for the cardiac abnormality. And of course, it gives tumors, as one would anticipate from the cells that are isolated from patients. But if you ask then Modelin's group or some other group, they just wrote an article in 2009, I think, it is sometime in the middle of the year. They wrote that KRJ is a better cell line and that bone is completely not good. So I'm not trying to confuse you. All I'm trying to say is that there is an intellectual and a rigorous process that goes on to know which is a good model to use. And so you can be rest assured that this goes on in every field, and it is not the only one. Every field is one in which the author of the original work will be very proud, promote it all around, but people around are going to do a lot of experiments with them and say, hey, but that's not right, or this is good. So the dust settles down, and in a few years' time, you know what's a good model. A lot of people will work with it, and it will be an accepted story. This is how the research goes. What do we do with these cells? So there are several ways in which you can put them in the mouse or animal and develop the tumor for the studies. So why mice? Because they are easy to do. I mean, there are a lot of they are inbred mice, so we know what is their genetic makeup. We know how they perform, how they behave, and a lot of studies have been done with this animal. So you can transplant or xenograft. So you could take a patient's tumor, a slice of a tumor, and put them in the mouse. You can do that. That's possible to do. You can do it on the surface. You can do it inside, or you can take out cells and then put them in the mouse. The mouse eventually will develop the tumor. There are certain restrictions. You can't put it in any mouse. So some mice, for example, normal mouse with normal immunological function will simply take your tumor cells and throw it out. It will be rejected. There will be no tumor. So we go around, circumvent this problem by having immune deficient mice. So there are very specific mice. We call them sometimes nude mice, if you have heard. Nude is without hair. So some of the models that I will show you, they are with nude mice. But these are the mice which will docilely take up all the cancer cells that you put, and over a few weeks' time, they will develop the tumor. So the second way of doing it is subcutaneous. This is the most popular model that has been used with all the cell lines that I showed you. It's a very simple idea. You just put the tumor cells on the back of the mouse, and over time, over a few weeks, you can see this little ball of tumor growing slowly, and then you do all the studies that you want to do with that tumor. The third way to do is intrasplenic injection. So this is another very cute idea that people have figured out, orthotopic liver model. Essentially, you pull out the spleen a little. I'll show you some photographs on how it is done. You inject the tumor cells in the spleen, and between spleen and liver, it's like a highway. 401. Straight, it goes in the liver. Within few minutes from spleen, the tumor will spread, the tumor cells will spread to liver. If they can take hold over there, you're going to get this nice liver tumor model, which you can do for your studies. It has been done since, it was done first in 1986, but I will not go into the history of it. Just suffice to say that this is, you put the tumor cells in one area and the tumor forms in another area. That's why orthotopic. And then there are the non-cellular animal models, such as serotonin-induced heart valvular abnormality. So some people who wanted to study just heart valve abnormality, they just kept on injecting serotonin in the mouse till they got cardiac abnormality. And so those are other kinds of models that are done. There are others that I will not go into, but drug-induced hypergastrenemia or Helicobacter pylori-induced gastremia. Intravenous model, for some strange reason, I mean, many other cancers that I deal with, IV is one of the standard. So you inject the tumor cells in the vein of the tail. For example, you can dip the tail of the mouse in hot water. The vein becomes really nice, visible. You can inject, and then eventually you will get the tumors forming either in gastrointestinal tract. You could get them in the lung. So with different tumors is done. For some reason, in this carcinoid field, it has not been done. Now, don't ask me why. Okay, so I will now give you a few examples of how things are done. So this is an example of how a subcutaneous tumor is done. So what we really do is we take this mouse has been injected with nothing or just PBS, that is a solvent, it's a saline, and this mouse has been injected with the carcinoid cells. This is the particular cell line that I use in this case is BON, and then the mouse that I use is biopsy nude mice. So each mouse, they cost money. That's why we need money for research. Each mouse costs about $120 just to buy. And then maintenance has to be in extremely safe condition because don't forget these are immune deficient mice. So they're going to get sick very soon, very easily. They will get infected very easily. So they have to be in very special HEPA filtered chambers. And it's like the ICU of mice. In the animal house, that cost me a bomb to keep one mouse in that. But they give the results. And those results you can see over here that at the end of 7 or 8 weeks, you're going to get this subcutaneous tumor. That's a nice tumor that's made. Why I'm showing you this model? Because these are the models in which you can do experiments. You can try to figure out how you can treat these tumors. This is the second model that I'll talk to you about. This is the intrasplenic or orthotopic liver model I told you. So what we really do is we take a very small incision in the mouse. This is mouse under anesthesia. We take a small incision, pull out the spleen, and inject it, and then suture the mouse back after injecting either the cancer cells or the control, where we don't inject anything. And then we wait for 8 weeks. And at the end of 8 weeks, this is the healthy liver that you will see in a control mouse. And this is the tumor that's developed. And in this particular example, a very badly developed tumor, very highly developed tumor, I would say, in liver. So these are the kind of models that one can use. Of course, when you develop this tumor in a mouse model, you need to know, is it really truly the representative tumor of the patient? And therefore, we have all kinds of analysis that we do, such as staining, and then we figure out whether this tumor is having appropriate serotonin levels that are being released in the patient's example or in this cell line. Chromogranin A one could do, or Ki67 parameter. So there are several ways in which you confirm that this tumor is really doing what it's supposed to be doing. Because before you start or work with any model, you need to be sure that's the model. Because if every result you get is with a wrong model, it has no value. So it's called quality control in every other field. This is the second aspect that we looked at, and this is part of the table that I took from my 2005 paper on how we discovered subclinical niacin deficiency in carcinoid syndrome patients, in some of them, 28% of the patients that we chose. Now, this particular study, which we published with about 120 patients, later on in a clinical analysis that was done and published in British Journal of Dermatology, they showed that actually if you take carcinoid syndrome patients and wait until they die, I mean, this happened to be the study in which they just followed them up, about 30% of those patients develop pellagra. Pellagra is an extreme niacin deficiency syndrome. So to develop pellagra, you have to be remaining niacin deficient for a long, long, long time. While the parameter that we chose was really the blood or circulating, we just took blood samples and we analyzed NAD or NADP, which is a a nerdy way of saying niacin level. And we just figured out that these patients are potentially subclinically niacin deficient. And this is the biochemical pathway why they are. So this is the serotonin that some of you know quite well about. So this is the serotonin, and you also heard about 5-HTP. So depending on what the patient is suffering from, whether it is a functional or non-functional, I won't go into those definitions, but either you will have serotonin or 5-HTP being made. Where is it coming from? It comes from what is at the top over here, tryptophan. So that's an amino acid that comes from proteins. And this amino acid, its normal job in everybody's body is not to make so much of serotonin. We all need serotonin, but not so much. So most of the time, tryptophan is really going to be degraded by a pathway which results in formation of NAD, which is what is derived from nicotinamide. So inside our cell, you need this molecule all the time. So this is his normal job. But in a carcinoid patient or in a carcinoid tumor, so much of tryptophan is going to go by the serotonin pathway, which will result in 5-HIAA urinary excretion or more of serotonin or more of 5-HTP, that they eventually suffer from niacin deficiency. And to answer this simple question, we actually did this analysis, and that was the work that we did in patients. So we asked the question, would it occur in my animal model? And yes, indeed, this is the liver. If we look at the NAD level, essentially what we are showing, instead of going into great details of it, that if you take the healthy portion of the liver, healthy, in inverted comma, healthy is what is not immediately affected by the tumor. But if you look at the tumor and the non-tumor portion, there is a distinct difference in the niacin level. So we know that this model is representing what is happening in patients. And then we asked the simple question, how can we improve this model? As you can see, I have to wait for 8 weeks to see the tumor. So we did the biblical, let there be light, and that's the light we added. So if all of you have seen fireflies in the evening when they go around with their little light looking for whatever they are looking, so the light has an advantage, is that that light is made actually by an enzyme called luciferase. Luciferase is an enzyme that uses energy, ATP, to a small extent, and it can generate the light. So you can imagine that that insect is so small, but firefly can go around making light as much as it wants. So, of course, scientists being what we are, we picked out that enzyme from the firefly, and then we can put that in variety of ways we do it. But essentially what we did was we put luciferase enzyme in a little DNA, which can then be put into carcinoid cells. So we put that into carcinoid cell. This is a cancer cell, carcinoid, in which we put the gene. And now we put this cell, modified, which is capable of making light, into the mouse. And now this is the model that we use normally. So all you have to do is, when the mouse is ready to be analyzed, you put this substrate, which is then broken down, luciferin, which is the material on which the enzyme acts and makes light. So how does it look? This is the machine which we use every week, every day. Our mouse has to go into this machine. They are anesthetized. They are alive. They don't get killed. Every week, one mouse, the photographs are taken of the mouse in this machine. And this is how the results would look. So you leave them under anesthesia, inject the luciferin, and we are going to be able to see, we are going to be able to see how the tumor looks in a live mouse. Week after week after week, we can see like that. So this is the point of injection. Within 1 week, I can see the tumor is coming up. And then eventually, as it grows larger and larger, and Dr. Oberg very nicely mentioned, this is the color scale. The red it looks, the worse it is, the bigger it is. And this is how the tumor will keep on growing, and we can monitor it without killing the mouse. And of course, we confirm it with the sacrifice at the end. So this is the necropsy data, and this is the live imaging that we did with luciferase. So you can see that if you see such a huge amount of red signal, you're going to get a big tumor. If you see a small signal like that, you're going to have this little tumor. Essentially, this is a model that we have made in which, without sacrificing a mouse, we can tell what's happening to the tumor that is progressing from what part of the body to the other part of the body, and that how big it has grown, is it responding to treatment. So these are the models in which we can do all the analysis. So now I'm going to show you just one data of unpublished work that we have. And Jonathan, the reason why I'm not giving more detail is that, as you can understand, researchers are very worried about burning their data. So We are hoping to be able to publish this work. But essentially, I will just give you an idea that we did a treatment X, and very soon we are writing a paper on this. But you can see that if a treatment is working, this is if without treatment, the controlled series of mice over the week, and this is after the treatment, what happens. So there is a significant suppression of the tumor made by the carcinoid cells in these mice. So this is how then you can quantify it by a variety of ways, either the light emission, or we are doing all the histological analysis on this tumor to find out. So the question, there are twofold questions. One is when you get the result, which we call this as a phenomenology, meaning we found an observation that this treatment X works. But more often than not, we are quite curious people, and we don't stop at this. We want to know why it worked. And therefore, we do all the mechanistic studies, and we try to find out what was affected. And that's how the studies are conducted. Jonathan, thank you very much for this. And now I will just tell what are the ways in which these models can be used. You will hear much more about next speaker, Dr. Izzat also is going to present the mouse model. But you can get a general idea how we can use this story. We can evaluate therapeutic options. We can do combination therapy. In my lab in particular, I'm doing first three things that you see over here. We are doing combination therapy, potentiation of therapy, and viral therapy. And I will show you some of the results. Sorry, not show you, but we have done some part of the gene therapy as well. And then you can do molecular characterization of the carcinoid with due care to be taken that, remember, this is a cell isolated from a cancer patient, from one patient. So if I want to generalize, I really need to get many more models coming from different cancer patients and then make a general statement that this is what happens in terms of molecular characterization. This has to be then tallied with all the histology and pathology data, which will be with most of the tumors that are collected from patient samples. And so this is the way the research can proceed. I think I will end this after giving you a brief idea of what I have done by thanking my collaborators. So I must thank Dr. Richard Warner of Carcinoid Cancer Foundation, who actually brought me to this field in 2001. He gave me a small grant of $25,000 to get going with his cancer patient samples. And then we roped in Dr. Jenny Spesieka of Calgary Health Region, and she was also very kind. She has a very nice neuroendocrine group that is over there, and they also ended up giving me about 30 or 40 patients' samples. Together we published the paper with the nutritionist from University of Guelph, not far from here, and currently the project is being funded and conducted with the collaboration from Jenny Spesieka and Grazia Graziani. And nothing is complete till the people who do actual bench work. I fly around everywhere, but I think it's these people who do the work. So I have to thank them. This is my team. Sometime middle of the year, I had taken the photographs, so you can see that some of those who are in red, they are the ones who have actually worked on carcinoid project in my team since last 7 or 8 years. Some of them, and I will now end with an example, if you will allow me, just 30 seconds, educational advantage and awareness of carcinoid among clinicians. It so happens that my lab works with carcinoid cancer and how do you do chemotherapy. So lots of MD students who are in Laval University are interested in doing fundamental research. And my lab is one where you can do from animal cancer to the molecular biology. So many of them want to work, and I'm very happy and very proud to be able to teach them. So many of them come for 3 months and practically stay on for a year doing part-time work in my lab, working on the carcinoid project. So one of these students, and it is Francis over here. Well, I have Francis, Mark, Matthew. I think many biblical names are there apart from that. I think it's more or less like a United Nations that is in my lab. So Francis was actually doing his internal medicine residency, and it was about 3 months ago. And he came and told me, Girish, you will not believe what happened today morning. And I said, yes, what? So they had a patient who was presented and they have an early morning 7:30 session on all the patients of the ward. And this patient had these strange symptoms of flushing and diarrhea and all kinds of parameters that everybody was floored. And of course, this is a teaching session for the residents. So the chief doctor asked, what do you think we should do? And then some of them came up with different ideas. And of course, they said, okay, it's a good idea, we should try it. And then Francis gets up and says, listen, I've been working on carcinoid in Girish's lab for the last few months. And I think that this to me suspiciously looks like carcinoid syndrome. And the chief said, come on, carcinoid, so rare. It is not a possibility. But nonetheless, since you raised the point, let's examine. And lo and behold, 1 week later, all the parameters, it was carcinoid, and now the patient is being treated. So you can imagine that education does play a great role, and I'm very happy to know that in small way we also contribute to that in our lab. Thank you very much with this. Thank you.
Thank you for a wonderful talk, Girish. That's great.